Dear students, in this session we are going to discuss about patterns of life in prokaryotes. What are the different categorization of prokaryotes? That is what we are going to discuss in this session. The various categorizations of prokaryotes. That means mainly bacteria. Yeah, the different types of categorization of bacteria. So the bacteria are categorized based on different criteria. So we are going to look into the categorizations or the classification. Not classification, we cannot say. This is not about taxonomy or classification. This is about categorization. So this prokaryotes or mainly the bacteria. Okay, prokaryotes here we are also discussing about archaea. That's why we are using the term prokaryotes. So mostly the organisms come here are the bacteria. But in certain categorizations here we see the presence of archaea also. Archaea as well as bacteria are prokaryotes, but they belong to two different domains. The ordinary true bacteria belong to the domain bacteria, and the other extremophiles, that is the bacteria or the prokaryotes which show extreme characteristic features. They will be having several differences in their bacterial cell wall as well as metabolism. And such prokaryotes are included under domain archaea. So both comes under this categorization. So here the prokaryotes are classified or they are categorized based on, there is a categorization based on nutrition, also categorization based on temperature preferences, that is at what temperature they exist, then categorization based on pH preferences, what the pH they require for their existence, which pH, whether it is neutral, whether it is alkaline or whether it is acidic, then categorization based on salt concentration requirements, whether they require higher salt concentration for their survival or they require lower salt concentration for their existence. In categorization based on oxygen requirements, whether they require oxygen or not. Some bacteria they live in anoxygenic conditions, means they do not require oxygen. In such cases, oxygen is like some toxin to this prokaryote. For anoxygenic prokaryotes, the oxygen may be acting like a toxin. So there are different types of groups of bacteria based on oxygen requirements. So here in this session we will focus on categorization of prokaryotes based on nutrition, and other categorizations we will be discussing in other sessions. So here categorization based on nutrition, here the bacteria or not bacteria, the prokaryotes, they are categorized into various groups based on the way in which they procure energy or make energy. That is energy. Food is actually energy. So how they synthesize this foodstuffs or the energy, based on that the prokaryotes are categorized into various groups. So already we know about the other organisms that they are classified into autotrophs and heterotrophs. Likewise, these prokaryotes are also classified or categorized into autotrophs and heterotrophs. Autotrophs, these are prokaryotes which can synthesize their own food from the raw materials, and the raw materials may be different, and depending upon the raw materials, these autotrophs are again classified into photoautotrophs and chemoautotrophs. Chemoautotrophs are also known as chemolithotrophs, and photoautotrophs are also known as photolithotrophs. It is referred to as photolithotrophs in certain textbooks and in certain journals. So these are prokaryotes which can synthesize their own food from raw materials. And these raw materials may be differing, and that's why it is categorized into photoautotrophs and chemoautotrophs. So what are these autotrophs altogether? These are organisms which can synthesize their own food. And the energy required for the production of these food materials,
Tap the banner.
the energy for the synthesis of this food may be obtained from light or from the breakdown of chemical compounds, and that is also included in this categorization of autotrophs into photoautotrophs and chemoautotrophs. Photoautotrophs obtain this energy from sunlight, and chemoautotrophs energy from the breakdown of chemical compounds. So that is the difference in the raw material here. Photoautotrophs use light, and chemoautotrophs use energy from the breakdown of chemical compounds. So about photoautotrophs, these are prokaryotes which can synthesize their own food from inorganic raw materials utilizing energy from sunlight. Okay, both photoautotrophs and chemoautotrophs use inorganic raw materials. They do not use organic raw materials. They use inorganic raw materials. But here the difference is photoautotrophs use energy from sunlight, and chemoautotrophs use energy from breakdown of inorganic chemicals or certain breakdown of some chemical compounds. So these photoautotrophs, they are having certain special intracellular pigments called bacteriochlorophyll. They are having certain intracellular pigments called bacteriochlorophyll which can absorb light energy. Okay, they can absorb light energy with the help of this bacteriochlorophyll, and it is converted into utilizable energy in the form of ATP. And this ATP is used for the synthesis of raw materials. And so here the plants also use this. In plants we do not find bacteriochlorophyll, we find chlorophyll in plants, and they use this chlorophyll to obtain light energy, and plants make their own food, synthesize their own food. But what is the difference between this photoautotrophs and photoautotrophs, which are, that is, plants, and this prokaryotes? Here this prokaryotes, they differ from higher plants and algae in that the bacteria cannot split water. In plants we can see they are splitting water for their hydrogen requirements, but here the bacteria cannot split water for hydrogen requirements, but they depend on chemicals such as hydrogen sulfide, NH3, for that, okay, for the requirement of hydrogen. And because of this they are not producing this water. They do not produce oxygen as byproduct. Okay, so they do not split water for hydrogen requirements. That is one major difference. Then another thing is that this bacteriochlorophyll, it is not put into discrete structures such as plastids or chloroplast. Okay, they are not placed in plastid or chloroplast. These bacteriochlorophylls are located in the inner regions of the plasma membrane. So these are two major differences from the higher plants and other algae. Of this higher algae, this prokaryotes, they also synthesize their own food like plants, but there are these differences. They do not use water. Then they are using bacteriochlorophyll. Then this bacteriochlorophyll is not placed into chloroplast. They are just attached to, or they are located on the inner regions of the plasma membrane. These are certain differences, major differences from higher plants. Then about chemoautotrophs, they also synthesize food from inorganic raw materials, but here they utilize the energy released from the breakdown of certain chemical compounds. Okay, these chemical compounds may be compounds of sulfur and nitrogen, such as ammonia, nitrates, sulfides. Okay, and this energy released is used for the synthesis of nutrients. Means when these chemical compounds are break down, the energy is released, and this energy is used to synthesize nutrients such as carbohydrates, lipids, proteins, nucleic acids from inorganic raw materials, that is from carbon dioxide and other nitrogenous chemicals. Okay, so simply we can say here chemoautotrophs, they synthesize food from inorganic raw materials using the energy released from the breakdown of chemical compounds such as ammonia, nitrates, sulfides, etc. Okay, they are known as chemoautotrophs. Then what are heterotrophs? When when we study about other animal and plant kingdom, there are not plant kingdom, animal kingdom, all of them are heterotrophs, means they depend upon other organisms for food. Animals exclude, only plants are considered as autotrophs in higher organisms, and all animals are considered as heterotrophs. And in heterotrophs itself there are these herbivores and carnivores, both are heterotrophs, they depend on other organisms for food. So here these heterotrophs, they depend on organic matter for their energy requirement. Organic matter, okay, means already synthesized food materials are used by these heterotrophs. They do not synthesize food from inorganic raw materials. They utilize organic materials, break down these organic materials, and the energy which is released from this breakdown of organic materials is utilized for their metabolic requirements. Okay, they are known as heterotrophs. And there are two types of heterotrophs, that is photoheterotrophs and chemoheterotrophs. So here about heterotrophs, which are prokaryotic heterotrophs, also they depend on organic chemical substances prepared by others for their energy requirements, and the energy released by the breakdown of this organic compounds, specifically the breakdown of carbon hydrogen bonds in the organic substances, is used for their physiological activities. Okay, and in most cases this oxidation or this oxidation of this organic compounds, it is not complete. Okay, energy is released, but it is not complete as in animals. It is not complete. It will, that reaction is not complete. That breakdown or that oxidation of organic molecules, it is not complete. So that instead of carbon dioxide and water forming, they produce certain intermediate products such as alcohol, lactic acid, citric acid, etc. Okay. In the case of higher animals, after the breakdown of these organic compounds, carbon dioxide and water is formed as byproduct, as the end product. But here this breakdown is not complete, or the oxidation of organic compounds, it is not complete, so that instead of carbon dioxide and water, intermediate compounds are formed such as alcohol, lactic acid, citric acid, and this process is known as fermentation. Fermentation, several bacteria undergo fermentation, and we utilize this fermentation for commercial production of several compounds like alcohol, lactic acid, citric acid, etc. So we humans are utilizing this property of bacteria for the production of so many commercial chemicals, commercially, for the production of so many chemicals commercially in large amounts. So we are producing that at industrial levels by utilizing this particular property of prokaryotes, that is fermentation. And so the heterotrophs can be further categorized into photoheterotrophs and chemoheterotrophs. This photoheterotrophs means they depend on light energy for deriving, that is they utilize light energy for the breakdown of organic compounds. They are known as photoheterotrophs. Examples are purple non-sulfur bacteria and green non-sulfur bacteria. What are chemoheterotrophs? They utilize organic compounds with the help of chemical energy. Okay, that is known as chemoheterotrophs. These are heterotrophic prokaryotes that obtain chemical energy by breaking down ready-made organic compounds. Examples are most bacteria, protozoans, fungi. So this is the categorization of heterotrophs, that is photoheterotrophs and chemoheterotrophs. So this is the overall classification based on overall categorization based on nutrition and energy capturing mechanisms. Microorganisms are classified into two: autotrophs and heterotrophs. Autotrophs into photoautotrophs and chemoautotrophs. Examples are given here. Photoautotrophs. Example: photosynthetic bacteria, green sulfur bacteria, purple sulfur bacteria, cyanobacteria, algae. Then chemoautotrophs: iron, sulfur, hydrogen and nitrifying bacteria. Iron bacteria, sulfur bacteria, hydrogen bacteria, nitrifying bacteria. Then heterotrophs: photoheterotrophs and chemoheterotrophs. Photoheterotrophs examples: purple non-sulfur bacteria, green non-sulfur bacteria. Then chemoheterotrophs example: most of the bacteria, protozoans, fungi. These are examples of chemoautotrophs. So this is an example of photoautotrophs. The first category, which are autotrophs. Okay, photoautotrophs: green sulfur bacteria. Okay, green sulfur bacteria. They are known as Chlorobiaceae. They are a family of obligately anaerobic phototrophic bacteria. They are anaerobic, means they do not require oxygen. Anaerobic bacteria. Obligately anaerobic means they do not require oxygen at all. And they are included under the phylum Chlorobi. You can see green sulfur bacteria in a Vinogradsky column. So green sulfur bacteria, they are non-motile. Okay, except there is an exception, chloroheptone thalassium, that which can glide. Okay, which they can move by gliding movements. So otherwise they are non-motile. Green sulfur bacteria are non-motile and they are capable of anoxygenic photosynthesis. Means they do not require oxygen. In the presence of oxygen they may die. They do not require the presence of oxygen. They are anoxygenic they are capable of anoxygenic photosynthesis. And in contrast to plants, what is the difference? In contrast to plants, green sulfur bacteria use sulfide ions as electron donors. Then they are found in depths of around 145 meter in the Black Sea with low light availability. And some of the characteristics of green sulfur bacteria: they use bacteria chlorophylls A plus, C, D or E. And location of this photosynthetic pigment is in chlorosomes or in the plasma membranes, located near to the plasma membrane. And photosynthetic electron donor H2, H2S or S, sulfur. And sulfur deposition is seen outside the cell. And what is its category of according to the nutrition? It is photolithoautotrophs. That is photoautotrophs itself. Photolithoautotrophs specifically, if we say. Then what is this chlorosome? I mentioned chlorosome in last slide. Means mentioned bacteria chlorophylls are placed in chlorosome or attached to the plasma membrane, located near the plasma membrane. So chlorosome is a photosynthetic light harvesting complex found in anoxygenic green bacteria. Okay, they are flattened ellipsoidal structures found in the cytoplasmic phase of the cell membrane, close to the cell membrane. Okay, the diagrammatic structure is shown here of that of chlorosome. Then this is chloroflexy. This is coming under heterotrophs, heterotrophic prokaryotes, heterotrophs. Chloroflexy. It is a group of green non-sulfur bacteria. They are photoheterotrophs. And these green non-sulfur bacteria, we say there are several groups of green non-sulfur bacteria and chloroflexy is one of them. So chloroflexia, they are typically filamentous. These green that is green non-sulfur bacteria, they are typically filamentous and they can move through bacterial gliding, and they are facultatively aerobic. Means they can live in the presence of oxygen or in the absence of oxygen. They are facultative aerobic. That's meant by facultative. They are not obligatory aerobic. They are facultative aerobic, means they can also live in the absence of oxygen or in the presence of oxygen. Then they do not produce oxygen in the process of this phototrophy, like plants. Okay, that we already mentioned earlier. They do not produce oxygen in a form of phototrophy because they are heterotrophs. So they do not produce oxygen. Then they have different methods of phototrophy, that is mainly photoheterotrophy. They have a different method of obtaining nutrition, that is photoheterotrophy. They are heterotrophs. Okay, they are different from that of true photosynthetic bacteria, and they do not produce oxygen also because they are heterotrophs, photoheterotrophs. So you can see the difference from phototrophs. We also explained one example of autotroph as well as one example of heterotroph. You can see the difference between these two bacteria. So about the other categorizations, all the other categorization, that is categorization based on temperature preference, then based on pH preference, based on salt concentration preference, oxygen requirement. In all these categorizations, we can find the presence of extremophiles, means bacteria that can exist in extreme conditions, extreme environmental conditions. And such categorization we will discuss in the coming sessions. Thank you.